Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on Lac Operon. Lac Operon is nothing but it is a kind of gene regulation system, students. So the first well-known gene regulation system was the Lac Operon. So the Lac Operon was discovered by two scientists, they are Jacob and Monod, in the year 1961. The Lac Operon controls the production of proteins needed for lactose metabolism in bacteria. Okay, students, let's see in detail what is Lac Operon. Okay, students, first you have to know what is operon concept. So, operon is nothing but group of genes, students. So, group of genes that function to produce protein needed by the cell. So here these are the structural genes, three genes. So the group of genes, so those three genes are transcribed at a time with the help of single promoter. So single promoter that initiated the transcription of a cluster of genes. Okay, cluster of genes are transcribed at one time. That is nothing but operon concept. So Lac Operon was first described operon. It is the best example for operon concept. Okay, students, here you can see the three genes. They are involved in the conversion of lactose, that is a disaccharide sugar, that is converted into two monosaccharide units such as glucose and galactose. So, three structural genes are transcribed at one time with the help of one promoter, that is nothing but operon concept. Here, three structural genes that produce protein, they are nothing but enzymes which are necessary for the breakdown of lactose. This is nothing but operon concept. So, if you see the diagrammatic representation of Lac Operon occupies 6,000 base pairs of DNA. So, if you see at the left side, Lac I gene is present. So, Lac I gene is nothing but it is a regulatory gene, okay, which is adjacent to the promoter region. So, this is the promoter region. Just upstream to the promoter region, there is another gene, that is regulatory gene. Okay, students. If you see the right side to the promoter, there is a region, nothing but operator region. After that, you will find three structural genes, nothing but Lac Z gene, Lac Y gene, Lac A gene. Okay, students? So, there are two different kinds of genes present in an operon. They are nothing but structural genes and regulatory genes. These three are the structural genes and this is the regulatory genes. Okay, students. So, these three structural genes, they are needed by the bacterial cell to break down the lactose sugar. They are grouped together on a single mRNA molecule. Okay, they are transcribed into a single mRNA molecule that is coded for the protein which is necessary for the lactose metabolism. You have to remember this. Let's see first three structural genes. Okay, these are the three structural genes they are going to express at one time. Okay, so first one is Lac Z gene, second one is Lac Y gene and third one is Lac A gene. Let's see these three genes. Okay, students. So let's see the roles of these three structural genes. Lac Z gene. Okay, students. So Lac Z gene that codes for an enzyme beta galactosidase. So actually it cleaves the lactose into the glucose and galactose. Okay students, what is the role of LacZ? It cleaves the lactose into glucose and galactose. So let's see the second gene, that is LacY gene. LacY gene that codes for beta galactoside permease. It transports the lactose into the cell. Okay, let's see the third one, that is LacA gene. LacA gene that codes for beta galactoside transacetylase. It is not essential for lactose metabolism, even though it appears to play an important role in the detoxification of compounds. Okay students, it involved in the detoxification. So it transfers an acetyl group from acetyl coenzyme A to the beta galactoside. Okay students, all these three enzymes, they are transcribed into a single mRNA molecule. So it should be needed for the lactose metabolism. It is an example for Operon concept. Why? Because here the three genes are under control of single promoter. You have to remember. Okay students, let's see the expression of these three genes. First, lactose enters into the bacterial cell through the cell membrane with the help of an enzyme permease. Okay, LacY gene expression facilitates the lactose into the bacterial cell. Now the LacZ gene will express. Okay, so LacZ gene produces beta galactosidase enzyme. It is nothing but lactase enzyme that breaks down lactose to the glucose and galactose. Okay students, so glucose and galactose, they enters into the metabolic pathways of bacteria. Then bacteria are going to utilize these two for the energy production. Okay students, this is the expression of these three genes. Now let's see the regulatory genes. If you see the left side to the promoter region, there you will find the regulatory genes. Okay students, they also code for proteins that controls the expression of other genes. Okay students, so it does not code for structural genes. Instead of structural genes, it produces a protein that is nothing but repressor or activator. Okay students, it could be either repressor or an activator. This regulatory protein will bind to the specific sequence, nothing but operator site or an activator site. Okay students, so actually this protein controls the transcription of structural genes. So if it is a repressor, it blocks the RNA polymerase and prevents the transcription. Okay, so if it is an activator, it helps RNA polymerase to bind to the promoter and increases the transcription. Okay? Okay students, regulatory gene also codes for proteins. They are nothing but regulatory proteins. It could be either activator or repressor. If it is an activator, so it binds upstream to the promoter region. It facilitates the RNA polymerase to do its work for the expression of Lac Operon students. So if it is not activator, if it is a repressor protein, that will bind to the operator region. Here you can see. So it is a repressor protein that binds to the operator region. It does not allows RNA polymerase to do its function. Actually, lac genes are not expressed in the bacterial cell regularly. Why? Because bacteria generally utilizes glucose. If glucose is absent, then only lac genes are expressed with the help of an activator or inducer. Okay, students? So, if the repressor protein bind on the operator region, then these three structural genes are not going to produced. Okay students, Lac gene is regulatory protein that produces repressor protein in absence of lactose. That repressor binds to the operator region and blocks the transcription of lac genes. If lactose is present in the medium, then what happens? So it acts as an inducer molecule that will bind to the repressor. Okay, students, causing it to detach from the operon. When lactose is binding to the repressor protein, so it is detached from the operator region. Then RNA polymerase transcribe the lac genes that is leading to the lactose utilization. So a regulatory gene that controls the expression of other genes by producing regulatory protein. It could be either repressor or activator that switch transcription off or transcription on. When the regulatory gene produces activators, then genes could be turned on. When the repressors are produced from the regulatory gene, then genes could be turned off. Okay, students? So, single mRNA molecule, it carries an entire operon. Okay, students? Here you have to remember one thing. Operon consists continuous structural genes. So, many genes, they are expressed with the help of single promoter that is present in prokaryotes only. It is absent in eukaryote students. Operon concept only present in prokaryotic cells. It is absent in eukaryotic cells. So, single mRNA that codes for many proteins. So that is nothing but polycistronic nature of prokaryotic genome. You have to remember. That is absent in eukaryotes. So eukaryotic genome is a monocistronic. You have to remember. Okay, students. So operon consists promoter region and an operator region. RNA polymerase will bind on promoter region. Here you can see. So promoter region is the site binding site for the RNA polymerase, whereas operator region is the binding site for the regulatory protein. Regulatory protein, nothing but repressor. So this is the site for repressor, this is the site for RNA polymerase. When repressor bind to the operator, then what happens? It blocks the transcription of these three structural genes. When operator is repressor free, then the gene will be produced. You have to remember, students. Okay students, let's see the exact mechanism of Lac Operon. Lactose is a sugar found in the milk. If it is present in the medium, E. coli needs to produce necessary enzymes to digest or to break down the lactose. Three different enzymes they are needed to digest the lactose or break down the lactose. These three genes are responsible for the breakdown of lactose. In the normal condition, that means when lactose is not found in the medium, these three genes are not going to be expressed because repressor protein will bind here. That is produced from the regulatory gene. It binds on operator region. It doesn't allows the transcription of these three genes. Okay, students. So repressor must be removed before the three genes transcribed. So allolactose is an isomer of lactose. It binds to the allosteric site of repressor protein. Here you can see lactose or allolactose that binds to the allosteric site of repressor protein. Hence, it inactivates the repressor. I have already mentioned that the repressor protein produced by regulatory gene. Okay, students. Lac Operon is an example of inducible operon because the structural genes are normally inactive. They are activated when inducer molecule, either lactose or allolactose, When it is induced, then only it will be expressed. That's why it is an example for inducible operon. Okay, students. Here you have to remember Lac promoter has two components. So that is, one is RNA polymerase binding site and second one is catabolite activator binding site. Okay, students. CAP, nothing but catabolite activator binding site, which is an activator protein. Binding of CAP to the promoter that activates the transcription of the Lac operon. Because CAP is a regulatory protein, it induces the Lac operon. When glucose is absent in the medium, you have to remember, CAP requires cyclic AMP to bind to the promoter. So, CAP requires CAMP. When CAMP is present at sufficient concentrations, okay, CAMP generally synthesized by an enzyme adenyl cyclase. CAP exerts positive control of the Lac operon. Positive control means lactose is going to be expressed. CAMP or cyclic AMP are synthesized by an enzyme activity adenyl cyclase. So CAMP is an activator molecule that should bind to the region which is adjacent to the promoter. Here you can see, when glucose is present in the medium, adenyl cyclase is inactive. It is not going to produce cyclic AMP. Then what happens? Cyclic AMP levels are low. Then CAP cannot bind to the Lac operon. Then what happens? Repressor protein binds on operator. It does not allow the transcription of Lac genes. Okay, students. If glucose is absent, then what happens? Adenyl cyclase become active. Then it synthesizes cyclic AMP. So, then cyclic AMP levels are high. It facilitates the CAP protein binds to the activator region. Then Lac structural genes can be induced. So, Lac operon requires presence of lactose and absence of glucose. In the absence of lactose, the Lac repressor binds to the Lac operator region. It prevents the RNA polymerase activity. Then 3 genes are not going to be produced.